So thank you for turning up for Blanche's confirmation of candidature seminar. For those of you who don't know Blanche, she did her honours in the School of Marine Tropical Biology on population genetics and conservation of narrow sawfish. And she she was going to do a PhD on sharks, but she saw the light that sea snakes are far more interesting than sharks, and so she's now going to be doing her PhD on sea snakes. She's supervised by me and Lynn Van Helden, Colin Simpendorfer and John Paul Hobbs in UWA. And her topic is West Australian sea snakes, an evaluation of their taxonomy, population connectivity, distribution, abundance and diet.
So, tropical marine ecosystems have been intensively studied, and what is really clear is that places like the Indo-West Pacific are characterised by extraordinarily high biodiversity and endemism, and the true biodiversity of even the most well-studied species, such as fish and corals, is not known, and that's becoming ever more clear with genetic studies revealing cryptic marine biodiversity on a regular basis. For example, Partridge et al. Took what was thought to be a widely distributed Indo-West Pacific species of mast grey, and their work revealed 7 new cryptic species. Similarly, there's examples from damselfish and flatheads. And this pattern is not restricted to fish or corals, it actually extends to sea snakes. 3 new species have been discovered from Australia in just the last 2 years. This species, Hydrophis donaldi, is entirely new to science, never been observed before. Apisurus mosaicus was thought to be a widely distributed Apisurus species, Apisurus eduxi. However, genetics revealed that there's a petition between Southeast Asia and Australia, with Australia containing its own endemic species. And there's a second instance of this, where a species in Australia that was thought to be Anhydrida schistosa has now been split off from the more northern parts of the Indo-Australian archipelago, and Australia again hosts its own cryptic species. Moreover, there's probably more cryptic species to come in the sea snake realm. This is a genetic tree from work by the Mochilegi for an Apisurus group species, Amitocephalus annulatus. And what you can see is these 2 samples down here from Shark Bay and Broome on the west coast of Australia. They're clearly genetically divergent from the animals from other regions, and it's possible that these represent new cryptic species in their own right. And there's quite a few cases of this in a number of taxa on the Western Australian coast of Australia. So, true sea snakes are an elapid subfamily Hydrophinae. So they're basically the evolutionary result of an Australian terrestrial elapid invading the marine environment. And together they comprise the most abundant and diverse group of marine reptiles on Earth, with 70 species in total. And the diversity of sea snakes is partitioned into 2 regions for 2 main lineages. So this is a map of species richness, and the numbers in the yellow bubbles represent the number of species found in a region. The Hydrophis group, which I'll clarify in the next slide, has the highest level of endemism in the more northern parts of the Indian Ocean and the West Pacific. In contrast, the Apisurus group has a high level of endemism focused in Australia. And you can see within Australia, with the sea snake biodiversity, that Western Australia has the highest diversity within Australia. So Western Australia is very special in terms of sea snakes. So the sea snakes petition into 2 main lineages. There's the Hydrophis lineage, the one from sort of the more northern parts of the Pacific and the Indian Ocean. This is a highly speciose lineage that's a result of a rapid recent adaptive radiation. And this group is predominantly associated with interreefal and soft bottom habitats, which tend to be more continuous in nature. In contrast, we have the Apisurus lineage, which is characterised by a few species and tend to be associated with reefs, so they're associated with a more patchy, discontinuous habitat. So we see a dichotomy emerging between the two main lineages, and this dichotomy is reflected in what's known about the genetics to date. So I'm going to give you a couple of examples using genetic work from Mochalehi. So this is a genetic tree. It's called a minimum spanning tree. Each circle represents a unique genetic haplotype. The size of the circle is proportional to the number of animals sharing that genetic haplotype, and the crossbars represent the number of base changes between each haplotype. And the colours represent the region from which the animal of that haplotype has. So this is Apisurus liber, which is a predominantly reef-associated but also interreefal species. And this is based on sampling from Northern Australia. And what you can see is that all of the samples from the Great Barrier Reef cluster together quite well. And the same is true of the Gulf and the West Coast. So this suggests really strong genetic structure with no sharing of genetic material between the two regions. And that makes sense because these guys have patchy aggregated distributions. It's also worth pointing out that these two animals were sampled as Apisurus libers, however they're highly divergent. One of these is another species, a restricted range endemic from WA, and another one of these is unknown to science and yet to be sampled further to work out exactly what it is. So if we switch now to an interreefal sample, an interreefal species, Lepimus curtis, we undertook sampling within Australia, and there's a lot of sharing of haplotypes between regions. So this one haplotype is shared by animals in the Gulf and the West Coast. So there's quite high gene flow within Australian waters. In contrast, when we look at the scale of Australia versus Thailand, we can see that there's absolutely no sharing of haplotypes between the two regions. So there's a strong barrier there that's also reflected between Australia and West Thailand. So what we can say about this is that whilst there's lots of genetic mixing within Australia, there's a large barrier to gene flow at the scale of countries. And I guess this highlights the fact that we really need species-specific knowledge for sea snakes because it's very different from group to group and species to species. And this pattern of continuity of habitat relative to the level of genetic structure is reflected in other groups. So we see it in sawfish. The freshwater sawfish has a reproductive cycle that's entirely reliant on fresh water. The pups can't survive outside of fresh water. And fresh water is a really discontinuous habitat. These guys also have a really inshore distribution, and that's reflected in the genetics where we see strong mitochondrial structure between regions within Australia. In contrast, we have narrow sawfish. Their pups live in estuaries. Estuaries tend to be connected by tracts of shallow water so things can move back and forth. And these guys have an inshore-offshore distribution out to 200 metres depth, so lots of continuous habitat. And again, it's reflected in the gene flow because we see low to moderate genetic structure in this species. So sea snakes, because we have these two closely related but distinct evolutionary lineages with differences in habitat use, actually form a really nice framework for evaluating the relationship between habitat use and genetic structure. Dietary specialisation can also be linked to genetic structure in order to evaluate vulnerability. So there's a really nice example where Lawton and colleagues looked at the chevron butterfly fish, which is an obligate corallivore that was thought to be really susceptible to declines in coral reef. However, they found that this species has really high gene flow across large geographic scales, so populations that, if they decline, they may be replenished from other regions, which ameliorates that vulnerability. So I would like to look at, so in terms of sea snakes, their secret ambush predators, secretive ambush predators, and it's really hard to observe what they're eating. And there has been quite a few studies to date, but they're low resolution because they're based on gut content, which is biased towards large things with calcareous body parts that don't digest easily. And there's also studies using isotopes, which are more general in resolution. Higher resolution and in a qualitative and quantitative context would be much better. And DNA-based diet analysis techniques actually offer this resolution. Sea snakes are really important within ecosystems. As an intermediate predator, the consequences of major declines in sea snakes can mean prey release for the things that sea snakes eat, and prey loss for the things that eat sea snakes, like sharks. And they're quite vulnerable to decline. They're live-bearing, so they give birth to live offspring at sea, and they have low fecundity, in some cases biannual reproduction. Additionally, they have long lifespans, so they may have long recovery times following declines. This, coupled with highly aggregated distributions and low dispersal rates in subspecies, is a recipe for vulnerability. And sea snakes are vulnerable to a whole bunch of things. The interreefal hydrophis lineage is highly susceptible to trawling and are frequently caught, as you can see here. In contrast, the Apisurus lineage, which is reef associated, is more susceptible to declines in coral reefs. All species are susceptible to loss and degradation of habitat, decline in water quality, and things like oil spills and seismic testing. So we've seen some really dramatic declines in sea snake in one particular location. And it's not surprising given the amount of threats they face. So Ashmore Reef off the west coast of Australia is a World Heritage Site, and one of its World Heritage values is the outstanding sea snake biodiversity of the region. However, between 1973 and 2010, sea snakes crashed by over 90%, and it's no longer a sea snake biodiversity hotspot because there's only 2 species left. And of the species that were lost, there's 2, Folia squama and Epifrontalis, that were only known from Ashmore Reef, so they've potentially gone extinct, which is of great concern. Additionally, there's an overall pattern of decline observed on the east coast of Australia at the Swains Reefs, which was work that was done by Mochilegi. In both cases, scientists can speculate about the causes of decline, but no one actually really knows. And clearly we're missing part of the picture. And the basic biology and ecology of most species is missing. So there's a really big opportunity there to address these knowledge gaps. Just to highlight the depauperate state of knowledge, this is the genetic sampling map for Apisurus laevis from V's work in her PhD. The purple stars represent the locations that she sampled. This is the most studied sea snake in Australia, and what you'll note is these black stars, which represent sampling gaps. So basically, almost nothing is known about the west coast of Australia. So if we look at distribution of species on the west coast of Australia, we have 3 Apisurus species. We have 2 restricted range endemics, the shark-based sea snake, here in yellow, the brown lion sea snake, here in pink, and then we have the broadly distributed species, Apisurus laevis. There's a lot of taxonomic confusion in this region, and often people say things like, we're going to dive with the sea snakes, with the olive sea snakes off of Broome. But the olive sea snakes off of Broome are actually brown lion sea snakes, and there's actually no evidence that Apisurus laevis occurs along the west coast. So it's pretty hard to manage the sea snake biodiversity if we don't actually know what it is, if we're not sure where the geographic ranges of the endemics start and finish. And furthermore, we have 2 broadly distributed Australopapuan species. We have Hydrophis elegans and Apisurus laevis. both of which occur down the west coast. These works actually revealed two really divergent mitochondrial haplotypes from the west coast, and it's possible that cryptic species exist in both these species. Which brings me to my objectives. The first one is to review the taxonomy and distribution of 4 species of sea snake between Shark Bay and Broome. My second objective is to compare the relationship between genetic structure and the continuity of habitat between the Hydrophis and the Apisurus groups using data that I would generate in my research. And then the third objective is to evaluate the extinction risk of restricted range versus widely distributed sea snakes. And this is particularly relevant for the Shark Bay and brown line sea snakes. The brown line is data deficient under the IUCN Red List, and the Shark Bay hasn't even been assessed yet. So I have 4 aims which become my data chapters. I'm going to use phylogenetics to evaluate the taxonomy and distribution of sea snakes, of coastal WA sea snakes. I'm going to use population genetics to look at population structure. I'm going to evaluate, more broadly, I'm going to evaluate the distribution abundance of sea snakes in WA using weighted remote underwater video station footage. And lastly, I'm going to use a really cutting-edge next-generation sequencing technique to evaluate the diet of sea snakes. So my thesis outline follows the 4 aims and includes a literature review on taxonomy and on the genetic structure of species in northern Australia. So there's a component of fieldwork in this project. I'm going to go out and catch sea snakes while scuba diving and snorkeling, so we use catch bags. I'm going to catch snakes from dip nets, using dip nets from boats, and I'm also going to collect dead snakes from shore bycatch. So, for the genetics chapters, I'm going to focus on 7 locations on the Western Australian coast. I may not get to all of these locations, but I will try my best, and it will depend on the availability of resources. I'm going to try to get up to 50 snakes per species per location. I do already have samples for olive sea snakes because I can access the Moxilades data. And I also have samples from Hydrophis elegans from the Gulf of Carpentaria and the east coast of Australia. So that's a good start for a genetic study. So to evaluate taxonomy and genetic structure, I'm going to use a comprehensive suite of 14 markers. I'm going to use mitochondrial DNA to look at female-mediated gene flow over evolutionary timescales, and I'm going to use nuclear microsatellites to look at gene flow over shorter ecological timeframes, and I'm going to undertake sequencing and genotyping to generate my data. For taxonomy, I'll just use phylogenetic techniques, a whole suite of standard maximum parsimony, maximum likelihood of Bayesian inference techniques. To look at genetic structure, I'm going to use analysis of molecular variants, discriminant analysis of principal components, and structure, which is a population assignment technique. To look at distribution and abundance, I have access to 7 years of BRUV data from 4 locations in WA that's been collected as part of long-term monitoring programs through AIMS, the University of Western Australia, the Commonwealth Government, and private entities. So I'm going to travel to WA to work in the BRUV lab over there and analyse the footage, and I'll calculate a standard indice, which is max N, is the maximum number of sea snakes observed in a screenshot at once to avoid resampling individuals that leave and enter the vision. I'll use a permanova with 5 factors to analyse the data, and then I'm going to use GIS techniques to map sea snake hotspots based on catch per unit effort. For the diet chapter, I'm going to focus on collecting dead animals from trawlers off of Shark Bay and Exmouth Gulf because I have existing collaborations there, and I'm also going to collect animals from as many other locations as possible so that I can look at intraspecific and intra-regional diet intraspecific diet on the West Coast, and then also, if possible, compare between the East Coast and the Gulf. And I have a lot of collaborations in place so that I can collect those animals on an ongoing basis, including trawl fishermen, the Department of Conservation and Fisheries in Western Australia, and also CSIRO. For the diet analysis, I'm going to use a technique called next generation amplicon sequencing. So, to give you a working example, Murray and colleagues studied penguins, and what they did was walked around on Perth Beach, picked up little penguins and put them in boxes with hot water bottles and waited till they defecated, and then they analysed that substrate. And it's such a powerful technique because they were able to identify 14 taxa in the diet and the relative contribution of each taxa. So this is just a few slides on how it works. I'm going to extract the intestinal content from at least 20 snakes per species per site from at least 2 sites. And then I'm going to use the polymerase chain reaction to amplify the genes of the prey. And I'm going to genetically tag each individual snake's prey so that I can then pool all of those products into one well for next generation sequencing, which saves thousands and thousands of dollars using the genetic tagging process. And then I'm going to send it for next gen sequencing. And from that, I'll get a library of about 7,000 reads per snake. And I'll use bioinformatic techniques, platforms such as Metagenome Analyzer, to match each of those reads to online databases like NCPI. If I don't get an exact match, I can use geographic information from things like FishBase to try and work out which species it is, or I can assign a higher taxonomic status. And then, quite simply, I calculate the percent contribution of each prey item. And then the last synthetic component of my thesis is to evaluate the extinction risk of the 4 nominal study species on the basis of IUCN criteria, and I'll also synthesise published literature in order to do that. So my publication schedule, in 2014 I will publish the BRUD data. In 2015 I'll publish 3 genetics paper focusing on restricted range endemics versus widespread species, and then a comparative phylogeography paper. And then in 2016 I'll publish the diet data. So this is my progress to date and my schedule. So I've been working on permits, and I've got my ethics permits, and I've got my WA permits, and I'm waiting for my Gurumpa permit. I am writing lots of funding applications, and I've undertaken training in next generation sequencing techniques and in venomous snake handling. In terms of my thesis completion, I'll travel to Western Australia fairly soon to start analysing the BRUD footage, and then be in the field by November to sample from Shark Bay to Exmouth Gulf. And then I'll do some lab work, get back into the field, but basically 2014 will be focusing on the 2 core genetics chapters, and 2015 will focus on the diet study. And then hopefully I will hand in November 2015. So I've budgeted the project at about $40,000, and I have $10,000 of that secured, $40,000 worth of funding applications pending, and lots of in-kind support in terms of field support with accommodation, access to boats and equipment. I'm being given the BRUD data set, and I'm also kindly being allowed to use the lab facilities of Murdoch University and the University of Western Australia. So I would like to thank my supervisors from Moksha Lady, Lynn, Colin and JP for working so hard with me. I'd like to thank some really great collaborators that have agreed to help me. Mike Hightouse from the Shark Bay Ecosystem Research Project, Mike Bunce, who is one of the leading experts in next-gen dietary analysis, and Alan Kendrick from WA Department of Environment and Conservation. And I'd also like to thank Gavin Creasel from Blackadder Reptiles, who gave me a free snake handling course, and Vinay for taking me out and teaching me how to handle sea snakes. Any questions?
Okay, so you've said that all these sea snakes have really cryptic diversity, and that it's hard to obviously look at them and tell what the difference between them is. So what are you going to do with your FRUB data to tell which is which? I'm
not going to be able to tell that. One of the important things that will come out of that is sea snake hotspots based on the Caterpillar depauperate, so that's going to be really important. With some of the interreefal species, because I won't just be studying those 4 core species, it'll be more broad, applying to everything I see. With some of the interreefal species, they're a little bit more obvious in terms of their morphology, but basically I'll just do the best I can and assign them perhaps a mysterious unknown.
Yeah. That seems reasonable. I was just wondering how you're going to do it.
Yeah.
It's obviously going to be a problem.
Yeah.
I'm also wondering about this sort of pattern of sea snake hot spots. And I'm wondering if maybe they move around, because it used to be, weren't just off, yeah, a couple islands, that's right, were really a hot spot, and I don't know if they still are.
It still is.
Oh, okay,
Yeah.
Okay. And so are they really going extinct, or are they moving? Is there any way we can discover what it is that causes a hot spot? For these guys, because it seems like it's completely mysterious why some places have heaps of. And
it is in these kind of data set from 35 years of data. It's basically this subset of reefs that change status. Most of them either stayed present or stayed absent, but a subset changed status, and it was from present to absent.
Mm-hmm. Oh, so that's very good.
So it's not like adjacent reefs were suddenly colonized.
It's really
so with olive sea snakes, they've done some translocation experiments, and I think they wouldn't even move 5 to 10 kilometres. Once you put them there, they stayed there. So for some species, it is changing more.
It's been a bit tired.
If it was interreefal species like elephants or it's different.
Yeah. Okay, so in that case, has anyone done any work on their disease? Because that's what happened with the frogs. People knew they were there, and then they just weren't there anymore.
I haven't come across anything in the literature.
Okay. So nobody's interested. Nobody's swabbing them. There isn't any. You haven't got anyone? It would be good to get some.
How people is trying to encourage people to do more physiology, and I guess that could lead to. People doing physiology are probably a bit more quick to do disease work. But yeah, it was a glaring gap.
Yeah. All right, I'll shut up.
Just out of curiosity, how easy is it to catch a sea snake? Since you're going to have to catch quite a few.
Yeah, they're a little bit less mobile than terrestrial venomous snakes, so they're a little bit easier to handle. And when you have them in the catch bags, you have safe zones on the edge of the catch bag, so you can handle them that way, and then you wear welding gloves to kind of hold them down, let their tail come out, and then take your genetic samples and then release them into a big crate for recovery, and then gently kind of let them leave. So you don't have to handle them except for the tail. Yeah.
In terms of the diet, are they dead snakes or are they live snakes?
They are dead. I would never kill a snake to do this study. So dead snakes come up into a bycatch, so I'll just collect them and cut them up. And I have a few samples from Weeper already that died.
How do you know the diet isn't seasonal?
It probably is, and it's not something I'm going to be able to. Well, if I can get enough samples, I'm going to be collecting samples from an ongoing basis, and I have no doubt that the diet's seasonal. If I can get temporal sampling, I will, but it just may not be possible within the scope of this project.
I was just wondering with your mitochondrial DNA, you said if it's going to give you an idea of the maternal gene flow.
Yes.
So do you have any feeling for what your microsate data will show? Are you going to show maybe that there's a final country, or is are males moving around more?
Yeah, I know from field observations suggest that perhaps male olive sea snakes move a bit more, but it's not entirely tested. And in general, for sea snakes, the microsat data has reflected the mitochondrial data. So I suspect them to be fairly similar. Lower structure in the Hydrophis group versus high structure in the Apatosaurus group in mitochondrial and nuclear markers.
Okay. Thanks, Blanche. I was just wondering that Rove's data, so you've got 7 years worth, and however many people you've got it from. Are you going to be watching the video?
So some poor little volunteers have been going through some of the data and cataloguing the location of the sea snake on the footage, so I just have to zoom to that location. And in some cases, they've actually been identifying the species to the best of their ability. So that saves a bit of time, but there is some data sets where I'm going to be zooming through hours of footage.
This is why you got minions. They don't have 4 months to
I was just going to ask, how come they have such cryptic diversity? Why do you think sea snakes were so hard to tell apart? Is it just because no one looks at them, or is it because of something else?
I think it's to do with historical sea level change. So with the rapid cycles of glaciation, and 6 of them in the last 100,000 years, we've seen this pattern of the Australian coastline having lots of shallow water and not a lot of shallow water. So I think the species have kind of come in and out and in and out, and there's been extinctions and radiations in that process. And coupled with the low genetic diversity, they've had the opportunity to, go through processes of genetic drift and lineage sorting, which is
How well tested is the technique to analyse the diet based on the genetics?
It's been done for quite a lot of taxa. It's been done in krill, in the Antarctic, sea lions, fish, other crustaceans. So it's pretty well tested. There are some inherent flaws, like biases. So if you have a particular prey species that has a mutation in the primer binding site, the primers might not bind and they might drop out completely, but it's still much higher resolution.
Has there been experimental validation of it?
There has. So this paper, Mariana, which is something that I didn't show, they used a more traditional DNA technique using cloning. And what they found comparing the cloning DNA technique versus the next gen, they found that the next gen had a lot higher resolution. And furthermore, when they pulled out the 4 most important diet species for the penguin, the relative contribution that they found in the next gen versus the cloning was comparable.
Yeah.
It was extremely similar. So it has been validated in that.
Yeah, I'm more thinking about an experimental study where you go and get a bunch of captured penguins and this particular thing and see if it works that way. Because I can imagine all sorts of biases in terms of the quantitative ability when you're looking at it following digestion.
Yeah, absolutely. I think the strength of it will be in the diversity of taxa that it pulls out, and what I'm really interested in a couple of things. I want to look at specificity of diet, because that links back to vulnerability, and something else that my brain just forgot.
But for the penguins, didn't they, it's been a long time since I read that paper, didn't they find the change in proportion of prey reflected the change in prey in the ocean?
Yeah, so they actually, following this study, they did temporal sampling, and what they found was a major shift in the prey assemblage of the penguins, and they actually think that was linked to a massive development in the core feeding area of those penguins. Yeah.
How do you think the weather's going to impact it?
Well, Shark Bay is a beautiful place. Colin tells me a lot of time is spent at the bar in Shark Bay.
Sounds good.
It's true.
Well, thank you. Very good seminar.
Thank